Okay, so we are working our way into gel electrophoresis now, into our organic section. So what we've done so far is we've done chromatography, and we did movement of molecules based on solubility as well as molecular mass. We then went through and did some practice loading of gels utilizing your micropipettes. So if you need a refresher on any of that information, make sure you check with me as we work our way into gel electrophoresis, into nucleic acids, into proteins, which is going to be that whole organic section that you guys are going to see. So with this gel electrophoresis, we're still doing separation of molecules. We're doing separation of DNA. We can separate out proteins. But now instead of solubility and molecular mass, now we're still doing molecular mass, size of the molecules. We're going to throw a little charge into it. So molecules are charged, especially your DNA and protein. Do they have a positive? Do they have a negative charge? So besides molecular mass, the separation will also be based upon charge. Um, Matrix was a word that we talked about before. It was one of the words that you guys were supposed to use in your introduction for your chromatography lab. And matrix within that chromatography was the chromatography paper. Matrix is what that molecule moves through. So in this particular lab, it is an agarose gel. It's very porous, just like your paper was. So that word agarose gel, just so you guys realize, you're going to hear these terms over and over again, but this ends in OSE. Anything that usually ends in OSE means that it's a sugar. So glucose, maltose, sucrose, agarose. It's some sort of carbohydrate, monosaccharide, disaccharide, could be a polysaccharide when it ends in OSE. And agarose gel, this matrix, actually is kind of this sugar substance that allows molecules to move through. It's a powder that I take, I dissolve it into buffer, which is kind of this electrolyte solution because I want to maintain a certain pH. Because of that pH of that solution, the buffer that these gels are in and your molecules are separating through, if it becomes too high or too low, it can denature or it can destroy the proteins as well as your nucleic acids, your DNA and RNA. So we're going to keep it slightly basic. We're going to keep it about 8.6. Remember, neutral is 7. So the buffer that we're using is about 8.6. Another reason we need this electrolyte solution or buffer is because it carries currents to it. It's kind of like a Gatorade solution. You can kind of think of it like that. So besides preventing pH change, it's going to allow us to carry the current. You guys are going to hook up to little voltage boxes and run a current through those particular gels so that molecules will be able to move. And because of the buffer that we have, the molecules are not going to denature, which means they're not going to unwind, they're not going to unravel, they're not going to become destroyed. It's the pH where we need it to be. It's going to mimic the pH of the conditions that they're used to. So if it becomes too acidic or too basic, proteins or DNA will denature. And that's one of the words that you guys are going to hear within this particular section. It just means that they get destroyed. So micropipette, you guys have used them, calibrated in microliters. Remember, it's 1/1 millionth of a liter. Here we are finally going to change the tips for each time we go in and out of a solution and in and out of the little wells or holes that you guys will be placing the solutions in. We want to make sure we wipe both sides of the pipette with a Kim wipes, like a little paper towel, so that we don't lose any of it in the buffer. We're going to try to keep the buffer as clear as possible. The wells itself, so let's pretend this rectangular structure is our agarose gel. I've molded and made little wells. There's a little plastic cone I put in as I'm making. It's kind of like Jello, and as it solidifies, I pull the cone out. And then there's these little openings here. You guys will see these little wells when you sit over the box, and you guys online will also see them in the little video that I'll be sending you. And it's going to make 8 lanes or 8 lanes within those wells for you guys to see. And you guys are going to be numbering them 1 through 8, but you have to be careful. There's always going to be a black end of the box as well as a red end of the box. And they'll be kind of color coded. There'll be a little black bolt on one side and a little red bolt on the other side. And we'll talk about these in a moment. But basically what's going to happen is that there'll be little wires that are going to run along the side of the box, along the gel, that's going to basically conduct the current. And those little electrodes are platinum wires. When you first turn on the box, this is the place that you're going to look. So right along the side here and along the red side, you are going to see these little platinum wires that are conducting electricity. Well, since it's in this buffer that's conducting current, you're going to see little bubbles floating. And that's one of the signs that this particular box is actually working. It looks like little soda bubbles when you pour soda into a glass. These platinum wires is basically what makes this gel box an expensive piece of equipment. So when I go through and make them, like I said, it's making Jello. If you've ever done it, I take a powder, I take some hot water, I throw it in the microwave, I let it boil. I use those pie pumps that we use for our spectrophotometry lab, and I start putting them into molds. And then I take this little cone and I place it in, and I can either do a mid-load well, where my openings are in the middle of the gel, or I can do an end load well. So I can kind of put them whichever way I want. So when you guys did your practice wells, I made you a whole bunch of, I put a whole bunch of cones in there so that you guys could get as much practice as you possibly could, because get rid of that nervousness and get your hands going so that you can physically load that without losing the sample that we need to do. So that comb is going to make the holes or depressions, which are called wells. When it solidifies, I kind of pop that comb out, I put the buffer in it, and we are good to go as soon as you guys load your samples in it. So why do we need two different types of wells? Well, your mid-load wells are for kind of like your unknown substances, proteins, enzymes. And the reason is because proteins, enzymes, or unknown substances have different charges to them. I don't know if they've got a positive charge. I don't know if they have a negative charge. So some might leave the well and go this direction. Some might leave the well and go this direction. If we're not sure, I want to do those mid-load wells. If I know what charge it has on it, such as DNA, I can put them all on one end and I know that my molecules are all going to be traveling to the other end. If I load proteins in here, some of my proteins, as they begin to break down to amino acids, some might travel this way, but I also might have some that go this way. And since there's no well for them or no gel for them to travel on, what happens is they actually will fall right off the well, go right into the buffer, and get destroyed, and we can't see them or utilize them. So this gel is in the gel box. It's under the buffer. It's called submarine loading, the way that you guys are loading, the way that you guys practiced before. Remember that the sample or the specimen that you're using, the liquid that you're using, proteins, unknown substance, enzymes, DNA, whatever sample you guys are loading into the well is more dense, it is heavier than the buffer that we're going in. So as soon as you start releasing that liquid from your pipette, it's going to sink wherever you are. So you want to make sure that that tip is slightly into the well of the gel. You don't want to go straight down into it, because what happens there is that pointed tip of your pipette will actually puncture. It's really soft. Puncture through the gel, and then you have it all coming out the bottom into that buffer. But if you go slightly into that little hole with the tip and then slowly release, first push, second push, pull completely out before you let go of your thumb, let go of the thumb, and then everything should sink down there. The problem is if you're not close enough, you could cross-contaminate your other wells, which we don't want to do. So we'll make sure that tip is slightly into those wells for me. Then we take that gel, it's placed in a gel box with our buffer, which we said is slightly basic, and it's going to help to have these molecules move. It's going to make sure that nothing's going to denature. And I said before, you guys are going to have to mark which end is the red end and which end is the black end of the box. And here's why. We said these things move based on charge. It's kind of like a car battery. That black end of the box has a negative charge to it. The red end of the box has a positive charge to it. So let's look at the black end of the box first. And you guys will be plugging in cords, and we have red cords and black cords. Black cord goes to the black end, red cord goes to the red end, goes into the voltage box the same way, black to black, red to red. That black end of the box, we said, is negatively charged. It is said to be the cathode end of your gel box. So opposites are going to attract here. So kind of like batteries, positives and negatives are going to attract. Kind of how we can create those bonds, with water molecules and everything, slightly positive, slightly negative, they're coming together and they are creating a bond. Same thing's happening here. Opposites are going to attract. So that black end of the box we said is negatively charged, which means molecules that are moving to that black end have a positive charge to them, because those positively charged molecules are going to the negatively charged end of the box. If that negative end of the box is called a cathode, my positively charged ions or molecules are called cations. And these are terms that you guys should have seen in your basic chem unit. I said cations, got the T in it, so positive. Those are my positively charged molecules. They are moving to the negative end because opposites attract. So we can say that your cations, positively charged, are moving to the cathode, negatively charged, and that is the black end of your box. So all these molecules are going to have charges to them. We also know they have mass to them. The closer they are to the wells, which is your origin, the heavier those molecules are. The farther they move away from the origin towards the end of your gels, the lighter they are in terms of molecular mass. Let's look at our red end. Our red end is our positive end. It is known as our anode. it will attract negatively charged molecules. They are known as anions. So my anions, negatively charged molecules, are going to the anode, which is positively charged. Opposites are going to attract. Anion has the N in it. It actually has two N's in it for negative. So cations got that T, anions got my N's, positive, negative. So that negative anion moves towards the positive anode. The key here is that opposites are going to attract. There's my gel box. So this is the entire structure. It is full of buffer. You'll actually see a red portion. It's not going to say positive charge for you, but you'll see a red dot on one end. You'll see a black dot on the other. So I know that this is my negative end. This is my positive end. There's my wells. They're also my origin. I will measure from the origin to how far my molecules move. If my molecules move towards the black end, I know they are positively charged. If my molecules move towards the red end, I know they are negatively charged. Opposites are going to attract. There are my wells, my origin. We said the farther they move. So I know that my pink molecule here is much, much lighter in terms of molecular mass than, let's say, my blue molecule. I know both blue and pink have positive charges to them. My yellow has a negative charge. It's about medium mass because it's in between a traveling distance from the origin between blue and pink there. Okay, so the heavier the mass, the closer they stay to the origin. So after loading the gel, we run the gel, we connect the electrodes, black to black, red to red, we turn the power supply on. I'll give you guys a number for all that. And eventually we're going to pull those gels. When it's completely done, I'll give you guys a time for that. It might be 20 minutes. We're going to turn everything off, unplug all the wires, and I'm physically going to pull the gel out of the box for you and put it on a piece of white paper. And just like chromatography, where you guys took those rulers and centimeters and you matched them up to the origin and you snapped the picture, we're going to do the same thing, but we're going to do two rulers. We're going to have one starting at 0, which is your origin, running towards the black end in centimeters, and one starting at 0 going towards the red end in centimeters. So we've got two ways that molecules can move. And that way you guys are going to be able to calculate things out. So I'm going to give you a little data chart on your lab. And RF value is still the same. It's still the distance each colored molecule moves. So think like each well is its own strip of chromatography paper. Well 1 might have 4 different colors in it. I might have 4 different RF values just for well 1. Well 2 might only have 1. Well 3 might have 3. You have a whole bunch more RF values to calculate out. Remember, there are no units. Distance each colored molecule moves in centimeters divided by the distance from the well to the end of the gel. And you need to measure both ways because the way that comb goes in, one way is going to be slightly longer than the other way, like a half a centimeter or so, but it's still going to make a difference. So you're going to have 2 solvent fronts, one from the wells to the black end, one from the wells to the red end. Okay. No units because that's a reference number. We're going to describe the molecule movements, describe them as a band. So you might see something like this. When you guys go to measure the origin and you put your rulers there, I don't care how you do it. If you're going to do it from the middle of it, if you're going to do it from the front of the well, from the back of the well, same thing with the colors. You're going to measure from where the color starts, from where it ends. You're going to measure from the middle. Keep it consistent however you are measuring it. So your little sketches on your lab are going to kind of look like this. Except I think I turned your gels the other way. I said, label for me which end is black, which end is red. Also, which end is positive, which end is negative. And you guys will do little sketches like this in both of your lab's mysterious moving molecules as well as your protein. Okay. You'll see a data chart. The first one, mysterious moving molecules. I will make you a data chart. The second one, it is all you guys. Okay, so make sure that you guys go through this. If you have questions before this lab starts, we can Google Meet anytime. Just let me know and we'll go through it. But this is something that you guys are going to be doing all throughout the year, not only with gel electrophoresis, but we'll also be doing something called ethidium bromide gels, which really don't utilize buffer. But we'll do that more when we do amplifying DNA, genetically modified foods, and we're going to amplify it and look at those genetically modified bands within those particular food samples. Okay. So with this, you guys will see our first lab, mysterious moving molecules. You guys may want to go ahead and kind of read through that before class so that you know what's going on and can ask questions, because it's going to be a really quick class for that. Okay, with this 39 minutes and a lot to do. Okay, let me know if you have any questions. Thanks.